Hello everyone. So today we are going to discuss on a topic called as transgenic plants. Transgenic plants are the plants which are unnatural plants. They are not formed naturally. So what is the importance of this transgenic plant? The technique of plant tissue culture along with genetic engineering has brought about a new, a plant variety. So these plant varieties are basically used for human benefit, irrespective it may be either for crop production, for some kind of diseases, for production of a new variety, a new gene pool. So irrespective, the ultimate aim of this transgenic plant is investors try to invest thousands of dollars or lakhs of rupees for this plant production, and the outcome what we have to get from this transgenic plant should be a novel kind of variety. So that is the key factor of any of the transgenic plant. So here we have some of the important transgenic plant. Herbicide resistant, you have pest resistant, we have increased shelf life plantlet, and we have new varieties which are produced. Now if you try to check the transgenic plants, they have different criterias. The plant should be a stable kind of plant, the plant should be having a market demand, or the product should have a greater demand in the market. So these are some of the criteria what we try to check. Apart from this, where all we have the application of this transgenic plant is with respect to agriculture, with respect to horticulture, with respect to industries, in forest. So where all we try to use plant products for human benefit. So there we go for transgenic plant production. And how exactly these transgenic plants are produced is by transgenic technique. We have biological mode of gene transfer technique, we have physical mode of gene transfer technique. We try to identify the genes, we try to insert the gene into the plant what we require, and the product what we are getting out of it, or the plant what we are getting out of it, that is called as transgenic plant. Now, transgenic plant with respect to agriculture. Now agriculture is a stream where we try to use the agricultural products. In this regards, how can a transgenic plant help in agriculture? We have two important examples here. The first one is herbicide resistant plant. And one such herbicide what we try to use here is a glyphosate. Now let us try to check what exactly the scheme is here. Shikimate 3-phosphate along with phosphoenol pyruvate. So this is compound 1, compound 2 are used to produce amino acids. Now what is the role of amino acids for plant growth? Now what is the role of this glyphosate? Glyphosate is a molecule, a herbicide, which tries to stop the production of amino acids. In turn they will try to kill the plant. Now take an example, I have a crop field. To this crop field, if I add glyphosate, irrespective of weed and a plant of my interest, both the plants would be killed. Yes, both the plants would be killed. Now what is my interest here is, I should either neutralize the activity of this glyphosate, or I should increase the activity of a particular enzyme. Now if we try to check here, glyphosate basically acts upon an enzyme called as EPSP. Now what is this EPSP? EPSP is enol pyruvyl shikimate phosphatase enzyme. Now as this glyphosate try to degrade EPSP. Once glyphosate inactivates EPSP, there is no formation of a complex. Once there is no formation of a complex, we have no amino acid which is produced. So that is the role of glyphosate. Now with respect to plant tissue culture and genetic engineering, we try to produce a transgenic plant which can degrade glyphosate, because we have seen many microbes including an E. coli has the capacity of breaking down this glyphosate. So what I try to do here is, I try to produce a plant. I try to take some of the genes from E. coli, try to incorporate the gene into a transgenic plant, and now I have two plant variety, a weed and a transgenic plant. I'll add glyphosate. What exactly happens? The weed would be killed, whereas the transgenic plant, because they have the genes from E. coli, they will try to break down the activity of glyphosate. So in simple, if there is no glyphosate, These two molecules combine with the involvement of enzyme EPSP. I have an active enzyme, that is active enzyme, where I try to get a complex. This complex in turn will try to produce amino acids. Amino acids are used in growth. Whereas wheat plant, same molecule, enzyme should be inactivated. There is no complex production. Once there is no complex produced, there is no amino acids and plants die. So that is one mechanism how we use transgenic plant in herbicide. Transgenic plant with respect to agriculture. The second what we have is pest resistance. Now one such example with respect to pest is American corn borer as well as tobacco bud. We have seen that a gene called as cry gene, which codes for a particular protein called as crystalline protein. Now cry gene is found in an organism called as Bacillus thuringiensis, a bacteria. So Bacillus thuringiensis has a gene called as cry gene. Cry gene produces crystalline protein. Now what is the activity of this crystalline protein is the feeding stage of any of the insect is the larva. So in larva stage they try to eat more and more so that they can grow. So by adding gene of my interest in the plant, cry gene, what happens is they try to produce a crystalline protein. This crystalline protein is inactive. And when do they get activated? Once they enter inside the midgut of larva. Now how exactly this will happen? As they feed on the plant, the crystalline protein enters into the larva. Once it enters into the larva, there is immediate change in the pH. High pH in the midgut will activate the inactive form of crystalline protein. So once activation of crystalline protein is done, they try to form pores on the midgut of larva. Once pores are formed and the midgut is damaged, obviously the organism is killed. This is how we have pest resistant plant which are produced by again PTC as well as genetic engineering. Now if you try to check here, there are two more examples when it comes to matter of horticulture. Horticulture is a kind of study, or it's a kind of subject where we basically go for plant varieties, something like flowers, increasing shelf life or flowers, or producing a new variety. Basically it deals with flowers. The first example what we have is increase the shelf life. Now what is the shelf life? Shelf life means a lifespan of a particular product. Now this product may be either flower or it can also be fruit. As my interest here is horticulture and I speak about flowers. Now we have a special gene called as NX gene. NX gene is a kind of gene which is obtained from, I've not mentioned, Pseudomonas species. Now what is the role of this NX gene is they degrade a complex called as aminocyclopropane 1 carboxylic acid. Now if you try to check, I have a molecule called as ethylene. Ethylene is a molecule which will try to soften the tissues irrespective fruit or flower. If at all it is a flower, it is at the carpel region. So ethylene biosynthesis always leads to softening the carpel. If carpel is softened, what happens is carpel just moves out or it gets detached. So my interest here is I should make sure that ethylene is not being produced. So what I try to do, take a gene from this particular bacteria. So as this is the biosynthesis process of ethylene, the product of an X gene will make sure it will degrade the complex called as aminocyclopropane 1 carboxylic acid. Now as I don't have this complex, ethylene is not produced. As ethylene is not produced, the increase in shelf life is observed. So the flower which was supposed to stay for a week, now it can stay say about 20 days. That is how increased shelf life happens by degrading the ethylene or the prior to that, the molecule of cyclopropane 1 carboxylic acid. I try to degrade this so that ethylene is not produced. Now we have another example. Production of a new variety. It's a special thing called as a blue rose. We might have seen white, we might have seen red, we might have seen pink. So the rose color, it's basically due to the process of dominant as well as recessive genes. So now scientists of Australia, what they have done, they have taken some genes from Petunia hybrida plant. As the genes are extracted from this plant, what they have done is the gene DLP gene codes for delphinidine protein. Now this delphinidine protein is a kind of anthocyanin. Anthocyanin are nothing but the colored or coloring molecules. So the product of DLP gene will give you a blue color. Now Australian researchers, what they have done is they have tried to isolate this gene called as DLP gene. They have inserted into a rose plant. Once they have inserted into a rose plant, what the product they have got out of it is a blue rose. It's a unique product. So unique product, an attractive product always has demand in the market. So in this regards, increase in shelf life as well as new varieties. This is just one example. Likewise, we have thousands of examples. Thousands of colored flowers can be produced. So this is how transgenic plant helps in horticulture. Semi-transgenic plant helps in agriculture. Thank you for watching the video.